Hey, 7th graders, today's assignment is the virtual photosynthesis lab, and this is going to kind of gear you guys up, hopefully, to a little bit of what we're going to be doing next week in lab hands-on. But you guys will be using a virtual link to a simulation where you will be manipulating some of the different reactants that are involved in photosynthesis to see how that affects the rate of photosynthesis. So in today's lab specifically, we're going to be messing around with the light source. Okay? All right, so a little introduction and background for you guys. This should be review for the most part. In photosynthesis, a plant takes in carbon dioxide from the air through its leaves and water, H2O, through its roots. It captures energy and uses this energy from the sun to convert the carbon dioxide and water into oxygen gas and glucose. So here's like the chemical formula or the chemical equation for photosynthesis. CO2 plus H2O and energy from the sun again reacts to form oxygen, O2 gas, plus glucose, C6H12O6. Using certain aquatic plants, scientists are able to see the rate of photosynthesis based on the number of oxygen bubbles produced by the plant. Today you will be manipulating your reactants to see how this affects the rate of photosynthesis and the production of oxygen and therefore glucose. So your procedure, you will change variables related to the light source to observe how much oxygen is produced in photosynthesis. This will also indicate how much photosynthesis is taking place and how much glucose is being made. For each variable changed, record the number of oxygen bubbles produced in 1 minute. Use the start-stop buttons on the timer. So right below our little light bulb here, you guys can right-click and open this link up, and you guys will see this little lab right here. It's nothing too fancy or crazy. And I'm just going to kind of show you guys through the 3 parts of the experiment how to collect data and how to answer some of the questions. So let's take a look at experiment 1 together. For each of these 3 experiments, you guys will start with a prediction. Okay, so for your first experiment, we're going to be changing the light intensity. And so you need to make a prediction. As you increase the intensity of the light, at this point I would assume all of you guys have changed a light bulb. And you guys have looked at the light bulb before and you've been able to see, all right, this is a 60-watt light bulb or a 100-watt or a 120-watt light bulb. Those kinds of things, that's the intensity of the light. So as you increase the intensity of the light, the amount of oxygen and glucose produced during photosynthesis you guys will go up here, you'll use your draw tool to either circle increase, decrease, or stay the same. So choose one of those 3 options. Okay, that is your prediction. We are going to test that result and see if you are right. All right, so moving on. Start with the 40-watt power source and observe for 1 minute. Reset and switch to 100-watt. Record the data in the table. So remember, you guys are counting the number of little oxygen bubbles you see in 1 minute time. So if you see a lot of oxygen bubbles, that means that light source is great. Photosynthesis is working very effectively, and you guys are producing a lot of oxygen and a lot of glucose or food for that plant. All right, so again, your 40-watt light source is right here, so we're going to make sure it's on 40. We're not going to do anything with the distance for this one. We're not going to change our light. We're going to stick with that white light, and then we're going to go ahead and hit start, and you guys are going to count 1 bubble, 2 bubbles. Notice the timer right here. When that gets up to a minute, you're done. 3 bubbles. And 4 bubbles, roughly. Okay? All right, and then you guys would go and you would change it to 100. Okay, and do the same thing. You guys are going to watch for a minute. And you're going to watch and count how many bubbles are coming up here. Okay? Once you guys have done that, you guys are going to record your bubbles in a minute for those time periods. And then you guys are going to review your results. As you increase the intensity of the light, the amount of oxygen and glucose produced during photosynthesis will. So look back at your data. Were you guys right in your prediction? Look at the numbers that you guys just came up with in this experiment. So go ahead and circle increase, decrease, or stay the same. Okay, you guys are ending up each experiment with this question. What is the independent variable or the manipulated variable in this experiment? That's the thing that you guys changed as scientists. So what did you change as a scientist? Was it the light intensity or the amount of oxygen produced? Circle one of those, okay? Second question, what is the dependent variable or the responding variable in this experiment? So I like to think of the dependent variable as the data that you're collecting. So it's kind of like a cause and effect. If you change this up here, what effect does that have down here? So what data are you collecting? What are you counting in this experiment? Okay, so that's your dependent variable. So circle either light intensity or oxygen produced. All right, for experiment 2, you guys are going to change distance from the light. So again, if I'm looking at this, you guys can go ahead and reset. So we're back to our normal things. We're going to try it at 50 centimeters and 100 centimeters. Again, you guys are going to time it for a minute. You'll count the number of bubbles, and then you guys will record that after you've made your prediction. So here's your prediction as you increase the distance from the light source, so you're moving the light source further away. The amount of oxygen and glucose produced during photosynthesis will increase, decrease, stay the same. Record your data in the data table, and then tell me how your results turned out. Okay? As you move that light source further away, did it actually increase, decrease, or stay the same? Okay, and then what is the independent? Remember, that's the thing you guys changed as scientists. So was that distance from the light source or the oxygen produced? And then what is your dependent variable? What is the thing that you are measuring to see if it changed? Okay, is that distance from the light source or oxygen produced? Okay, experiment 3, you're going to look at light color. So a little bit different way of phrasing our prediction. When you change the color of light between white light, red light, green light, or blue light, they also call white light clear light. What light color will be the most effective for photosynthesis? So choose one of those 4 light colors for your prediction. And then same thing in here, you guys are going to reset, but for your lab today, you guys are going to run the experiment using the 100-watt bulb and 50 centimeters away. So make sure you're 100 watt here, 50 centimeters away, and then you guys can use these 4 filters here, time them for a minute, record your data. And if you guys look too, if you guys run this down here and you hit start, and then once your minute is up, you guys hit stop, your guys' data will also record right here. So you guys can kind of do this without having to count, and you guys can just kind of watch the timer and say, all right, I stopped it here. And you guys should be able to see, oh, never mind, I lied. It doesn't do that for you guys. Sorry, I wasted your time. Okay, anyway, record your guys' number down here. Make sure, again, you guys use the start and stop. And then after you guys collect your data for this, I want you guys to take a look at this graph. Okay, so this graph shows us how chlorophyll in leaves absorbs lights. So on your y-axis here, you guys have your percent absorption. So if we're absorbing a lot of light, generally we should be doing a lot of photosynthesis. So you guys can see of the light that hits that, this color is almost 100% absorption, versus down here we have our spectrum. It shows us our different wavelengths based on the color here. You guys can see which lights are most effective and which are most absorbed in a plant or in chlorophyll found in the plant leaves. Okay, so with that being said, from your results, which light color produced the most oxygen bubbles? So look back at your data table, tell me which light color produced the most or had the most photosynthesis, and does it match the graph? So does it match the ones here that we had the most photosynthesis happening, or the most absorption of light? And then explain your answer. From your results, which light color produced the least amount of oxygen bubbles? Does this also match the graph? Explain. So again, look back at the graph. And then finally, your wrap up. Do you have any questions? Circle yes or no. If you have some, type them below. And then 4, 3, 2, 1, how do you think you did? Great job. I hope you guys had fun with this, and hopefully we'll be looking forward to doing it hands-on next week. I'll talk to you soon.